Well, welcome to this webcast. We appreciate your attending it. I'm very excited to be here, joined by Dr. Dirk Scheinert and Dr. Muller-Holzbach. The topic of the presentation today is going to be Revolutionizing Your Practice, New Data Which Shapes the Era of Drug-Elution. As most people know, peripheral arterial disease is manifest in multiple different ways. Above the knee, it's mixed morphology with about 50% chronic total occlusions, lots of calcification, can be challenging anatomy as a result of those things, and medium to large size vessels ranging between 4 and 7 to 8 millimeters in size. Below the knee, calcium predominates, and in critical limb patients, it's very difficult to recanalize some of these vessels due to angulation, especially the anterior tibial artery. These tend to be relatively small vessels, making working in them difficult. And the occlusions that occur there are very long and accompanied by calcification and multiple branch vessels, making recanalization a challenge as well. In this slide, you can see the multiple angiographic demonstrations of the difficulty and lesion characteristics that many of us will encounter. The first here is calcification, easily identified by the filling defects and stenoses created by them. And the second is in-stent restenosis, which when it occurs can be very difficult to engage, clear, and maintain patency in. Third is the chronic total occlusion I mentioned before, and especially when accompanied by calcification can be difficult to cross or in fact re-enter. And then lastly, thrombus, which can be difficult because of the potential for distal embolization and the voluminous nature of this in some cases. As we look at the growing burden of PAD around the world, we see that in fact in high-income countries it has been present for some time, not surprisingly given diet and other excesses in the styles of living. But in low-income countries, as they come online and as nutrition increases and smoking becomes more available, we see that there's a rapid increase in PAD over the last decade. And worldwide, PAD has increased by almost 1/4 from its previous levels in 2000. If we look at the global treatment of PAD, in fact, there's some differentiation among the different areas, and you can see this slide is differentiated by the United States, the European Union, Asia-Pacific countries, and South America. And I think probably most prominent is the fact that about 50% of patients in the United States are taken care of by endovascular means, and a relatively small number of them, 20%, are treated with medication only, or I should say non-exercise medication only. The remainder are treated by bypass surgery and exercise. But we see in the remaining countries actually a dominance of medical therapy, which really is suggests that most patients aren't offered a revascularization approach. And in fact, even in the patients who are offered a revascularization approach, only the minority of them will receive something like bypass surgery or endovascular means. So there's a lot of opportunity here for patients worldwide to improve their outcomes and to improve their functional capacity. And now I'm going to hand it over to Dirk Scheinert, who's going to talk to us about the interim data from the Ranger SFA trial. Dirk?
Well, thank you very much, Bill. It's a great pleasure for me to share with you today some of the interim data from the Ranger drug-coated balloon trial in the superficial femoral artery. Well, these are my disclosures. I'm a consultant for Boston Scientific. I guess most of you are familiar with the Ranger balloon. It is what we call a next generation drug-coated balloon technology. The device was CE marked in July 2014. It's basically built on the Sterling 018 platform and uses the so-called transpac coating, which contains paclitaxel together with an incipient which controls the coating integrity. The device is available in a wide size matrix for femoral and for below-the-knee applications, as shown here on the slide. Well, specifically about the transpac coating, you can see it's, as I said, it uses paclitaxel as the active agent, and it uses a citrate ester as the excipient, which is basically designed to balance the hydrophilic and hydrophobic properties It allows the adhesion of the balloon to the balloon surface during the transit time to the lesion, and then it controls the efficient drug delivery to the vessel wall. And most importantly, it's a very stable coating which is designed to really minimize the particulate loss during the whole process. Well, these are some preclinical tests which were done with the coating, also comparing the device with other devices on the market. And you can see here that this hydration test basically resembles what happens during the transit of the device to the lesion. And importantly, it was shown that the Transpex coating remained fully adherent to the balloon during the hydration. So it basically shows that really only very minimal drug can be lost during the transit to the lesion. This is another preclinical test, which is very important, I guess, which looked at the coating integrity and showed deliberated particles which were kind of filtered from the circulation. And clearly here you can see that the Ranger balloon has a very low particle loss during this whole process. Well, this slide gives you an overview on the ongoing DCB clinical trial program with the Ranger. As I said, I will today update you on some of the preliminary data from the Ranger first-in-man study. There are other trials ongoing, like the DCB Ranger project in China, which is a single-arm 123 patient study. And then there is the DCB SFA global pivotal trial, which is going to be conducted in the United States and in other geographies. Well, as said, the Ranger SFA study was set up as a first-in-man study. It's a randomized trial with a 2-to-1 randomization of the Ranger drug-coated balloon versus uncoated balloon. It was conducted at 11 sites in Germany, France, and Austria, and was looking at a target enrollment number of 105 patients. And actually, the trial is just about to close enrollment these days. The primary endpoint of this trial is very similar to other first-in-man trials, was in-segment late lumen loss based on an angiographic follow-up at 6 months. Well, this is basically the currently available data set for the interim safety analysis, which was carried out in August. You can see that at this time, 93 patients had the procedure, and about 65 patients were past the 6 months follow-up time window, and there were about 42 patients out to the 12 months time window. Here you can see some of the patient characteristics. Well, I think we can see it's very well balanced. Of note, the percentage of diabetic patients was in the range of between 33 and 40% in both groups. So very comparable, I think, to other trials which have used similar patient cohorts and similar approaches. You can see here the lesion lengths. I would say rather long for a first-in-man study, with about between a lesion length between 7 and 8 centimeters, as shown here in the first line. And otherwise, you can see that most patients were treated for lesions in the middle or distal superficial femoral artery. Of course, most interesting at this moment is the situation of occurrence of serious adverse events during the follow-up time window, and we can say that to date, no serious adverse events, no death or major amputations were reported. The only events which were really of interest were the target lesion revascularizations, and we had actually 8 TLRs in the control group and only 5 TLRs out to 6 months in the DCB group. And if you remember that it was a 2-to-1 enrollment, it becomes already visible that here the rate in the DCB group was quite a bit lower than in the control group. Overall, the freedom from target lesion revascularization at 6 months was 89.1% in the DCB arm. Well, of course, these are very early data and also preliminary data, but still, if you project these freedom from TLR, these TLR rates for the Ranger study, here shown in the right columns, you can see that there was a good difference in TLR rates with 11% in the DCB arm compared to 32% in the control arm in this preliminary analysis. And that compares very favorably, I think, to previously reported data with other devices and similar trial designs. Well, even if we want to go even further in predicting potential trial outcomes, we can look at the time course of occurrence of TLRs. You can see that if you look at these events displayed here in this time curve, we can see that there is a tendency that TLR, freedom from TLR curves would separate over time, potentially showing a difference, then an even bigger difference at the 12 months follow-up time point if that remains consistent. Well, I think it's important to say that these data are very encouraging, however, of course, preliminary. In parallel to these activities, we can say that there are a number of physician-initiated research activities which basically focus on a variety of disease states, looking at hemodialysis, grafts, and fistulars, which, as we all know, have very high recurrence rates. There are trials set up to look deeper into the superficial femoral artery region and also into the very challenging below the knee area. So I think in conclusion, we can say that this new generation DCB technology, the Ranger balloon, has achieved an optimal transfer rate and tissue retention at a very low loading dose, just using 2 microgram per square millimeter paclitaxel. And importantly, preclinical tests have shown not only the efficacy but also the safety and stability of the coating with really very low particulate formulation and drug loss during transit time. The Ranger interim safety analysis shows in a clearly preliminary fashion that so far there are no safety concerns, no major amputations or death. And interestingly, it gives a signal that we can expect a higher TLR-free rate with the DCB, with the Ranger DCB, as compared to the plain balloon in angioplasty. And well, as I told you, we are confident that we are going to see that there might be even a stronger separation of these TLR curves going further into the 12 months follow-up. I think the Boston Scientific Drug-Coated Balloon Clinical Trial Program is a very robust series of studies, both the major studies initiated by the manufacturer, but there's also a series of physician-initiated studies looking at very important disease states, including hemodialysis access, SFA applications, and below the knee, which I guess is going to give us a lot of insights into the clinical utility of this new technology. So I would like to thank you very much for your attention, and I hand over to Stefan Müller-Hülsbeck, who's going to continue. Thank you.
Thank you, Dirk. I'm now honored to present the Majestic Trial 12-month results evaluating the Eluvia Drug-Eluting stent in the femoral popliteal artery. The study objective is very clear. We wanted to determine whether the Boston Scientific nitinol drug-eluting stent, the Eluvia stent, shows an acceptable performance at 9 months when treating the SFA and or the popliteal artery lesions up to 110 millimeter in length. The Eluvia stent system is an SFA drug-eluting stent with a biostable polymer and paclitaxel combination, and it's built on the well-known Eluvia stent platform. It's a laser-cut self-expanding nitinol stent with a triaxial delivery system. Looking a little bit closer to the Eluvia coating design, I have to say it's a so-called dual-layer system consisting of a conformal coating for both layers, the primer layer, which promotes the adhesion of active layer to stent, and the active layer, meaning the paclitaxel, and this controls the release of paclitaxel. The biology of restenotic cascade in the SFA is in this context very, very important, because we know that smooth muscle cell proliferation can continue for up 100 days or longer after stent implantation, and extracellular matrix formation can continue well beyond 300 days. Restenosis following nitinol stenting in the SFA peaks therefore at around 12 months. If we now look at the sustained drug release and looking at drug release over time, comparing the drug release behavior of the silver PTX and the drug-eluting Eluvia stent, then we can see that the major difference, because drug release from the Eluvia stent system is sustained over time. That means over 90% of drug is released at 1 year. Drug release coincides with the restenotic cascade, and this is something I want to highlight at this point. Looking a little bit closer to coating design comparisons, I have to say that, for example, the silver PTX has no carrier, whereas the Eluvia drug-eluting system has proven promos polymer. And there's also a major difference in terms of the drug dose. The silver PTX has a drug dose of 3 microgram per square millimeters, whereas the Eluvia stent has 0.1657 micrograms per square millimeter. The size matrix is quite similar, but I want to highlight that the stent length of the Eluvia system is with 150 millimeter, 30 millimeter longer than the silver PTX size matrix in terms of length. And if you look at the scanning electron micrographs at a magnification of 100, you see the uniform distribution of the polymer and the drug on the Eluvia stent, as you can see in the right corner. The Boston Scientific peripheral bare metal stent and drug-eluting stent clinical program consists of the Supernova trial. It's a bare metal stent trial, a so-called prospective multi-center single-arm open-label trial, which was done in several centers in Japan, in Canada, in Europe, and in the US, including 299 patients. The drug-eluting stent trial, the so-called Majestic trial, is also a prospective multi-center single-arm open-label trial, which included 57 patients in Australia, Europe, and New Zealand. And the program will be completed by the Imperial drug-eluting stent trial. It's a prospective multi-center trial with a randomized controlled 2-to-1 comparison, comparing the Eluvia stent with the silver PTX stent in round about 500 patients. And this kind of trial will take place in several countries worldwide. Now I would like to focus on the Majestic study design. It's a prospective single-arm multi-center trial. As I mentioned earlier, we included 57 subjects in 40 centers in Europe, and the primary endpoint was primary patency at 9 months after stent placement, and the follow-up through 3 years was planned. The enrollment criteria were as follows: chronic symptomatic lower limb ischemia, defined as Rutherford categories 2, 3, and 4, de novo or restenotic lesions. The degree of stenosis was greater than 70%. The vessel diameter should range between 4 and 6 millimeter, and the lesion length should range between 30 and 110 millimeter. If you look a little bit closer at the baseline patient characteristics, you see we have the typical characteristics. We treated older patients, mainly male gender, and the patients suffered from typical risk factors like smoking, diabetes, hyperlipidemia, hypertension. They had cardiac history and so on, and definitely they had a peripheral vascular history that was the reason for treatment. If we look at the baseline lesion characteristics, I want to highlight that we mainly treated the distal SFA. We included also the proximal popliteal artery up to a length close to 71 millimeters, and I want to point out that most of the lesions, 64.9%, were severely calcified, and 46% of the lesions were total occlusions. We met the endpoints. At 9 months, we reached a primary patency rate of 94.4%, and the additional efficacy endpoint with a 9-month TLR rate of 3.6% was also reached. Now the 12-month data, due to the primary patency, we achieved a very high rate with 96.1%, and this results in a Kaplan-Meier estimate up to 96.4% primary patency at 12 months with the drug-eluting Eluvia stent. The safety profile is as follows. We had a 3.8% 12-month major adverse event rate, and this consisted of no death, no major amputation, but 2 target lesion revascularizations with 3.8%, as you can see here. I also want to say that the stent integrity was guaranteed because no stent fractures upon angiographic core analysis was seen. If you look at the patient outcome, and this is obviously of utmost importance for the patients, I have to say that 94% of the patients were classified as Rutherford category 0 or 1 at 12 months, and ABI improvement sustained through 12 months also. So the conclusions I want to make based on the Eluvia data are as follows. The Eluvia dual-layer coating design allows for a sustained drug release that coincides with the restenotic cascade. The MAJESTIC study of the drug-eluting Eluvia stent in the femoral arteries met its primary efficacy endpoint with a 9-month primary patency rate of 94.4%. Primary patency was sustained through 12 months with a rate of 96.1%, accompanied by a TLR rate of 3.8% and 0 stent fractures through 12 months. Last but not least, patients exhibited symptomatic and hemodynamic improvement through 12 months. Thank you very much. And now I would like to hand over to Dr. Gray.
Well, thank you, Stefan. I'm pleased to be able to talk now about the implications for our practice of these new and exciting therapies, really. Here are my disclosures. So your life on drugs, where do they fit into our practice? They may be considered standalone therapies in many patients, either drug-eluting stent or drug-coated balloon. But we have lots of patients who would not have fit into the trials that you've just heard about or are anticipated. These are more complex patients with task C and D lesions, heavily calcified lesions, and patients with in-stent restenosis, none of whom were included in the trials to date. We also don't know how drug elution in combination might work. And lastly, and probably most interestingly, we have lots of opportunity for adjunctive therapy in drug elution, where atherectomy, a plaque tack, or shock wave type of calcium disruption may be actually quite interesting, and how drug-coated balloon or drug-eluting stents work in those combination therapies, unclear. You can see in this slide that SFA lesion complexity drives the therapeutic testing, that is, the trials that have been done to date, but actually not patient selection. Because actually when we talk about the trials that have been done, it's been relatively simple lesions, relatively short, not heavily calcified. But we use these stents, drug-eluting balloons and drug-coated balloons and drug-eluting stents in many more complex lesions where we don't really have a lot of data. So this, I think, is a gap in our knowledge base. We do, as you probably have already heard, have a multiplicity of drug-coated balloons which are available. These are just 4. There are probably double or triple this number in the EU. And without going through each one of them, I do want to just highlight some of the differences between them. The first is in the dosing. You can see that for the most part, most drug-coated balloons will have a 2 microgram per millimeter squared service dose. The difference there is in the Impact Admiral balloon, which has a 3.5 microgram per millimeter squared. The second most interesting difference is the coating technology, the excipient. The excipient is a carrier which allows for the drug to mix, become crystalline on the balloon, but also leave the balloon and enter the vessel. And I'm not going to go through all of them here, but each one of these balloon formulations has a different excipient, all meant to act as a spacer and to provide this function of drug delivery at the vessel site. And then lastly, there are some differences in the sizing matrix which is available. In the Boston Ranger, there will actually be BTK sizes from 2 to 4 millimeters up to 150 millimeters, which is unique among the drug-coated balloon matrices available. When we look at drug-eluting stents, it's a little simpler. There are really only two to talk about. There have been several failures in drug-eluting stents, but these are the two that have really emerged as winners. The Zilver PTX, which is already available in the United States and in Europe. It's based on a Zilver platform, and it has no polymeric coating, which that means the paclitaxel is just sprayed directly onto the balloon, onto the stent. In the Eluvia version of drug-eluting stent from Boston Scientific, it doesn't have approval just yet, but it's based on the Innova platform. It's also nitinol, but its difference is that there is actually a bioabsorbable polymeric matrix which will control the release of the paclitaxel into the vessel. The differences in the size matrix are not too significant, other than the fact that Eluvia will have 150 millimeter size. So if we start thinking about how to use these drug elution technologies, the first would be as a standalone treatment algorithm. So in our lab, we will pre-dilate a lesion, which we think is potentially appropriate for a drug-coated balloon. We'll make a decision after the pre-dilation as to whether we think the vessel will stand on its own or whether there's enough residual stenosis, dissection, or recoil that it may need a scaffold or potentially a second balloon dilation. Once we've made that decision, we'll go with a drug-coated balloon if we think the result can stand alone, or we'll use a scaffold like a DES for residual stenosis, recoil, and dissection. In our lab, we actually don't treat a patient that doesn't get an antiproliferative therapy. We believe strongly in this new mode of treatment. But what if they fall outside? What if the lesion falls outside of the kind of relatively simple lesion and has calcium? I'll take calcium on as a first example. We know that calcium leads to lots of different mechanisms of vessel failure after PTA. The first can be a late recoil or acute dissection and recoil. The second is under expansion or limited expansion, and even with stent, you may get compression and lack of full expansion. And we know now that through some very interesting work by Dr. Finelli, that calcium probably limits the ability of drug elution to actually exert its full effect. So in the Finelli study, he looked at 60 patients with SFA stenosis or occlusion, and he did very excellent imaging, very careful imaging, to look to see what the quantity of calcification burden was, and then looked at 1 year and correlated the calcification burden with the outcomes. And you can see that by this graph that as the calcification quadrants fill up, and as you get to a full calcification circumferential nature, that you see lower patency, lower ankle brachial indices and patient outcomes, and greater late lumen loss and TLR rates, all of which suggest that the drug effect is somehow diminished by the presence of circumferential calcium. So the question then becomes, well, we have technologies that potentially could modify calcification. Is there actually benefit to modifying calcium with these technologies? And the one that we have most readily available is atherectomy. It clearly increases luminal diameter. It decreases burden of plaque. It removes or modifies calcification. And in fact, there's some recent data from Elazar Edelman's lab that suggests that in cadaveric models, the degree of drug uptake is about 50% increased after orbital atherectomy in his model, suggesting that, in fact, not only modification but uptake may be important. The next difficult patient population is the in-stent restenosis. It's very difficult to treat. Restenosis rates after treating in-stent restenosis range to 70%. And excimer laser has been shown to debulk and improve outcomes, but still has a 50% 6-month restenosis. Coronary ISR has been successfully treated with DCB, And the Zilver Global Registry suggests that there's good effect with drug-eluting stent. And I put up here the 6-month, actually the longer data, looking at both primary patency as well as TLR rates at 6 months, improved over what historical controls would otherwise suggest in the global registry data. When we as operators, when we look at these data and we try to compare across trials, it becomes difficult to do that because there are some drivers of outcomes among these trials that we might be confounded by trial to trial. So some trials may contain more diabetes, more longer lesion length, more total occlusions, or worse calcification. And in this slide, you can see that we've lined up by rank order all the trials, many of the relevant trials. And you can see that the rank order is by lesion length. And if you follow the various bouncing balls and bars here, you can see that the calcification and chronic total occlusion rates can be different among these trials, making cross-comparison of the trials very difficult to do with any satisfying outcome. I think you contextualize the outcomes here, but direct comparison is very difficult. So now we have an exciting glimpse into the future. I'd like to talk briefly about the Boston Scientific DES Global Pivotal Study, the Imperial Trial. This is a trial, a randomized trial, which will compare the Eluvia drug-eluting stent to a Zilver PTX stent in the treatment of superficial femoral artery and/or popliteal arteries. I'm privileged to be a principal investigator with Dr. Muller-Hosbach, and the objectives of the trial are to demonstrate both the safety and effectiveness of the Eluvia drug-eluting stent, the vascular platform, in treating SFA and popliteal disease in lesions up to 140 millimeters in length. The study design is a good one. It's a prospective multicenter 2-to-1 randomization, Eluvia versus PTX, and a controlled single-blind non-inferiority trial. There'll be a concurrent non-blinded, non-randomized single-arm trial looking at the pharmacokinetics of these stents. If we look at the subjects, there'll be almost 500 subjects with over 300 treated with Eluvia and about 150 treated with the Zilver PTX. And then there'll be 8 to 12 subjects in the PK study. The investigational centers will range to 75 centers worldwide throughout US, Canada, New Zealand, Belgium, Germany, Austria, and Japan. And about 5 centers in the United States will be in the PK study. The primary efficacy endpoint will be primary vessel patency as assessed by duplex ultrasound at 12 months and adjudicated by an independent core lab. And the primary safety endpoint is fairly standard, all-cause death through 1 month, target limb and major amputation, and target lesion revascularization through 12 months. So in conclusion, there's a vast number of endovascular therapies used to treat PAD. Drug-elution therapies represent an improvement over many of the non-proliferative approaches, but have been developed in a limited, relatively simple lesion set. Comparing the study outcomes from various devices is difficult due to the complex nature of the PAD being treated trial to trial. Data on using drug-elution technologies in combination with other adjunctive therapies needs further investigation. And lastly, the Imperial study will provide the first head-to-head prospective multicenter data comparing to an existing drug-elution therapy and should further help in clinical decision-making. Thank you very much. I'd like to pass it over to our operators, and I appreciate your time.